Okay, so next we have our second player from Team Langebio. That's our postdoc, Sergio Nigenda-Morales, who is jointly appointed in my lab, but together also in the bioinformatics lab from the Calle Abreu in Langebio. So briefly, I want to say that Sergio is our stellar biologist in the lab, so everything has to do with non-humans, we go and ask him. So he also has multiple projects in multiple species, but I think today he's going to focus on the opossum data, which is awesome. Yeah. So thank you, Sergio.
Okay, yeah, awesome possum. So thank you very much for staying here. I'll jump right in because I have very few time. So as probably you know, most of the marsupials in the Americas are currently distributed and have evolved in South America. But very few species have actually moved into North America, and one of those species is the Virginia possum, or in Espanol, tlacuache or zarigüeya. And it has been estimated that the ancestor of these species with molecular data, they estimated that probably crossed to Central and North America around 4 to 7 million years ago. And the fossil record indicates that this species was already present in southern North America, Texas and Florida, by 1 million to 0.6 million years ago. And then during the last 15,000 years after the last glacial maximum, it colonized the rest of its range till northern Canada. So as a result, we have a species with a really wide range, going from from Costa Rica till southern Canada. And it has many different phenotypes along this range. So the objective of this study, of this talk, is to estimate the population structure and genomic diversity and identify what the probable demographic scenarios have shaped this diversity in this species. So to do that, we collected 186 samples through several locations along the distribution of this species, and we did a genotype by sequence approach. And at the end of the day, after filtering a lot of SNPs, genotypes, and individuals, we ended up analyzing 20,000 SNPs and in 127 Virginia opossum individuals. So the population structure or mixture analysis find the most support for 4 main groups. The United States group, all the populations there, the central western Mexico, southern Mexico, and Cozumel. You can see these 4 groups also in a PCA analysis. Here, US, central Mexico, western, southern Mexico, and Cozumel. But as we can see here, there is some substructure to that we can see better with a PCA analysis. In the US, we have California, Indiana, South Carolina, and probably here the East Coast. So we have 3 to 4 populations there. Actually in the state of Morelos alone, we have the tlacuaches from Cuernavaca and Sierra de Huautla and Jalisco here. And when we analyze the southern Mexico, Cozumel is very different. And when we analyze southern Mexico without Cozumel, we have this nice shape of the Yucatan Peninsula here with Quintana Roo, Yucatan, and Chiapas. So we ended up with around 11 populations. So when we look at the FST values and we compare the FST values between these populations, we observe that if you remember the 4 main groups, populations within those groups have very low FST values. Right here, northern populations, central western Mexico, southern Mexico. But when you compare these populations against the other main groups, the FST values are quite high. And notably, but not surprisingly, the Cozumel Island population is kind of the most different from everyone else. And when we look at the genomic diversity, namely heterozygosity and nucleotide diversity, we see that the northern population have lower diversity in general, and also Cozumel, which is not surprising. So we're probably talking about a bottleneck here, the island. And the rest of the Mexican populations have higher diversity. So so with this data, it kind of fits a northward expansion, right? With northern populations with less diversity because they recently been funded or they yeah, so they've been funded very recently, and Mexican populations have longer time to develop the diversity. However, when we look at the phylogenetic tree, we see these 4 very clear groups again, but it doesn't fit a northern expansion because the northern populations diverge first. Under a northern expansion scenario, you will expect the northern populations from the United States to diverge more recently, right? So, so that kind of contradictory results. When we see the Tajimas D, we see clearly in the Cozumel Island kind of a bottleneck, a positive Tajimas D, and the rest is not very clear because they are not super negative, right? It might be expansion in all of them, and from California and the East Coast population, we don't have enough data to have a Tajimas D value. So what we think it explains better our data is more than a northwards expansion, is more a glacial refugia in which during the last glaciations, in the well, not the last glaciations, but during the older glaciations, Pleistocene and Pliocene, one small population of Virginia opossums were probably trapped in the southern part of the United States and remained isolated from the rest of the populations in a really small place, well, in a really small refugia, maintaining small population sizes, and it only expanded recently after the last glacial maximum. And so that explains the early divergence of those populations in the north, the reduced genetic variation, because just recently expanded, they were really reduced, probably in incipient structure, and recent divergence among them. So these data, these results, they expanded recently. So the importance of these results is for the evolutionary history of opossums that previously was thought it was a northern expansion, but apparently it wasn't like that. So we have to test actually this hypothesis. And for the Cozumel Island population is very interesting because apparently it's very divergent, so it could have some conservation implications there. So I want to thank my previous lab, my lab now, and if you want to talk more, we have other, as Andres said, other projects with whole exomes and whole genomes. And if you want to check out my research, and so there are my information, just drop me an old email. So thank you.
All right, thank you, Sergio. Questions? We're running out of time, so maybe you don't say yes in 1, 2, 3. Just kidding, just. Okay, great. Okay, go ahead.
That's a really good question. I didn't want to actually say that, but when we look at this tree, I mean, it's here is very divergent, so I don't know. I mean, these 4 groups, they fit different subspecies that from morphology have been defined, except Cozumel. So that wasn't actually ever, like, it was part of the Yucatan group, according to morphological studies. But this was surprising for me, and it's really old, apparently. We have to formally test it and kind of get some divergence time here. But I'm leaning towards that, but I cannot say it now. I don't feel comfortable saying it. Yeah.
Well, you said it. Okay. So just in the interest of time, I would suggest that we move on, but thank you, Sergio.
Thank you.